Welcome to the deep dive. Today, we're tackling something really critical in how your medications get made safely. We're talking about how scientists find, control, and manage those unwanted extras, impurities, and byproducts that can show up when making drugs. It's all about making sure the medicine is pure and safe. We'll be exploring the strategies, the analysis, the whole detective work behind it.
Exactly. And our mission, you could say, is to distill the key ideas from various sources. We're looking at, development guides, regulatory thinking, especially around tricky substances, and even some real-world case studies. The goal is to give everyone a clear picture of this complex area, but without getting totally bogged down in the jargon.
Okay, let's start at the beginning then. When a drug first shows promise, the amounts are tiny, right? Lab scale. But moving towards clinical trials, towards patients, that means scaling up big time. From milligrams maybe to kilograms. And our sources point out this scale-up step, that's often where the impurity challenges really surface.
Yeah, it's interesting, isn't it? Making more of something isn't always straightforward. Bigger batches can actually open the door for new side reactions. It might seem a bit counterintuitive, like think about baking a giant cake versus a cupcake. Heat distribution changes. Ingredients interact differently over longer times. Process chemists are the experts here. Their job is designing synthetic routes that aren't just scalable, but also cleaner by design. They try to avoid toxic stuff and, maximize the good product.
And there's one type of impurity that gets a lot of focus. You sometimes hear about them, genotoxic impurities or GTIs. These could be things like alkylating agents, maybe nitrosamines. The worry is their potential to, well, damage DNA.
That's the core concern.
Yeah.
They can interact with our DNA, which is obviously not good. And if you look at the wider regulatory landscape, like the thinking behind the ICH guidelines, there's a huge emphasis on assessing and controlling these specific DNA-reactive impurities. These guidelines are basically the international rulebook for drug safety. M7, for instance, really drills down on how to handle these potential mutagens. So drug developers, right from the start, they need to figure out if their drug's specific chemistry poses any unique risks, like forming GTIs. And they need a solid plan for managing those risks all the way through development and manufacturing, which is exactly why you need those sharp analytical techniques early on just to even spot these potential problems.
Okay, so impurities can pop up, especially when you make more stuff. But what are the actual strategies? How do scientists try to keep them low from the get-go? It can't just be about cleaning up afterwards, right? There must be proactive approaches.
Oh, absolutely. A big part of it is cleverly modifying the chemical process itself, designing it so it naturally produces fewer byproducts. We saw a great example in one case study on dicomatinib synthesis. They actually tweaked the synthetic route, the steps involved. And by doing that, they significantly cut down on a specific unwanted impurity, abyssamide. It's about smarter chemistry design up front.
Right, so it's less like mopping up spills and more like designing better plumbing so it doesn't leak in the first place.
That's a perfect analogy. Process research is exactly that, developing synthesis methods that work well at large scale and address potential problems, like impurity formation, before they happen. Another source mentioned a synthesis for azacrominone and atacromilamine. The initial way they tried making it hit bottlenecks, which could have led to more impurities. So the solution wasn't just tweaking, it was devising a whole new way to assemble the molecule, a different strategy altogether. And often, especially later in development, you see a shift away from really complex or, let's say, inefficient chemical steps. Things like multiple oxidation state changes. The idea to streamline the process, make it more efficient, and crucially, less likely to generate unwanted side products. That decomitinib example also sort of touched on this optimization aspect.
And it sounds like even something seemingly basic, like just the recipe, how much of each chemical you add, the stoichiometry that plays a big role too.
A massive role. One paper we looked at detailed the synthesis of a particular intermediate chemical. It showed how critical it is to control the exact amounts, the stoichiometry. If you don't, you risk getting overreacted products or underreacted ones. Both are impurities. Think about baking again. Too much sugar, different cake, not enough flour, it falls apart. Same idea in chemistry. An imbalance leads to side reactions, leads to impurities.
Okay, so scientists are trying hard to prevent impurities during synthesis, but inevitably some might still be there, maybe in tiny amounts. That's where the analytical chemists come in, the detectives. How do they actually find and measure these trace unwanted things?
Yes, this stage is absolutely vital. The sources really stress using a strategic, chemistry-informed approach. It's not random testing. You use a whole toolkit of analytical techniques, often step by step. Like forensic tools at a crime scene, each tells you something different. HPLC, mass spectrometry, NMR. And these techniques help differentiate. They help figure out which impurities are, let's say, relevant, maybe they're above a certain threshold or known to be risky, and which are just potential impurities, maybe detectable, but at such low levels they aren't likely to cause harm. So a potential impurity might just be a tiny blip, well below any level of concern, but a relevant one. That needs careful monitoring and control based on safety data and regulatory limits.
So it's not just about finding every single other molecule, but really focusing on what could actually matter for safety or effectiveness.
Precisely. And having that deep knowledge, knowing what impurities could form, also helps make other important decisions, like choosing the right kind of packaging or figuring out the best long-term storage conditions. You need to know if an impurity might increase over time, affecting the drug's stability or potency.
We mentioned genotoxic impurities earlier, GTIs, and the sources really underscore why they're such a big deal. Can we explore that a bit more? What makes them particularly concerning?
The fundamental issue, as we touched on, is their potential to damage DNA. And DNA damage, unfortunately, is linked to cancer risk. That's the main concern. So regulatory guidelines, the thinking reflected in ICH standards, puts a very, very strong focus on these. For the really potent ones, sometimes called the cohort of concern, things like aflatoxin-like structures and nitrosyl compounds, alkylisoxy compounds, the acceptable levels are incredibly low. We're talking parts per million or even less. The acceptable daily intake is likely to be vanishingly small. Ideally, you want zero presence, especially since they usually offer no benefit to making the drug anyway.
That makes total sense. Why have something potentially harmful around if it's not even necessary for the process?
Exactly right. Now, there can be very rare situations, maybe for a serious disease with few or no other treatments. A covert mutagenic impurity might be, technically, unavoidable in the synthesis. In those specific rare cases, the principle is as low as reasonably practicable, a LARP. This means you have to prove you've done absolutely everything technically feasible to minimize it, even if you can't eliminate it. It requires a huge amount of scientific justification, data, and usually close discussion with the regulatory agencies. It becomes a very careful risk-benefit calculation.
Now, listening to all this identifying, preventing, detecting, controlling it feels like there must be an enormous amount of paperwork, of record keeping involved. Our sources didn't explicitly detail process documentation, but it seems like a really robust system for tracking everything is just essential, a kind of paper trail for the medicine.
You've absolutely nailed a critical point there. While the specific term process documentation might not have been highlighted in the snippets we looked at, the whole framework of good manufacturing practices, GMP, definitely implies it. It underpins everything. GMP is the bedrock ensuring quality and consistency in all pharmaceuticals. And a core part of GMP is traceability. Being able to know exactly how every batch was made, what went into it, what checks were done at every single step that's fundamental for managing impurities. It lets manufacturers and regulators trace back any potential issue to its source and make sure corrective actions are taken effectively.
Right, so it's about having that clear, auditable history. Proof that every stage was controlled properly to minimize contamination and ensure the final product is pure.
Precisely.
Yeah.
GMP requires very tightly controlled manufacturing conditions, validated processes, everything designed to prevent unintended substances getting in or forming. And this ties directly back to that ultimate goal we keep mentioning, making sure the medicine that gets to the patient is safe, pure, and does what it's supposed to do, effective.
It really does all circle back to the patient in the end, doesn't it? All
Yeah.
these complex strategies, the detailed chemistry, the high-tech analysis, the strict rules. Ultimately, it's all geared towards ensuring that when someone takes a medication, they can trust it. Trust it to help, not harm.
That really is the fundamental ethical principle driving all this.
Yeah.
The absolute goal is delivering pharmaceuticals that not only work therapeutically, but meet the very highest standards of quality and safety for the people who rely on them.
Okay, so to kind of wrap up our deep dive today, we've journeyed through this really crucial world of impurity management in making medicines. We've seen how vital early identification and proactive control are, how important it is to optimize the chemical synthesis itself to avoid making byproducts. We've touched on the indispensable role of sophisticated analytical techniques for finding and understanding impurities, even tiny traces. And we highlighted those specific worries around genotoxic impurities and the really strict controls needed there, all under that umbrella of good manufacturing practices and ultimately patient safety.
And thinking ahead, maybe a final thought for our listeners. As drug molecules get more complex and manufacturing itself evolves, How much more sophisticated will these strategies and tools need to become? What role will continuous innovation, new analytical methods, maybe even AI, play in ensuring drug quality and safety in the future? It's a field that's constantly moving, constantly adapting to protect public health.